Do you think we can start now?
Yes, fine.
Okay. Okay, so welcome everybody to this month's. Oh, I'm sorry about that. That should not have happened. All right, so welcome everybody to this month's CiNet Junior Scientist Council's pop-up webinar series. Today we have with us Dr. Dipankar Dwivedi. Dr. Dwivedi is a research scientist in the geochemistry department at Lawrence Berkeley National Laboratory. Dr. Dwivedi received his PhD from Texas A&M University, and he has a strong background in flow and reactive transport modeling, watershed hydrology, biogeochemistry, and machine learning. Dr. Dwivedi's research focuses on developing a predictive understanding of climate-induced ecosystem responses across spatiotemporal scales in mountainous and coastal wetlands. So with that, I would like to welcome Dr. Dwivedi, and you can feel free to share your screen and the floor is yours.
Thank you, Tanya, for a nice introduction. Let me share my screen here. Okay. Can you see my screen?
Yes, looks good.
Okay. So yeah, thank you for the background, and I'm very happy to give a talk here, and thanks for the opportunity. So today I will talk about a scale adaptive framework to predict water and nutrient fluxes across land-water interfaces. And I will also talk about like why we need a scale adaptive framework. So to motivate that, I am going to also talk about like some high fidelity reactive transport modeling work. And those actually motivate to develop like scale adaptive framework and probably hybrid modeling where we can also use machine learning. But before I start, actually, so I want to take this opportunity to promote two events. So one is like HydroML Symposium that is being hosted by Lawrence Berkeley National Laboratory. And I think a lot of you might already know, but if you do not know, so please be part of this symposium. And Praveen is also on the organizing committee, and Tarun, I know he's going to be here in person. So although deadline has passed, but we haven't started reviewing the abstract, so you can actually reach out to me. And second thing, because we are talking about critical zone research here, so I want to draw your attention. So we have a special issue on in Frontiers in Water. So on investigating connectivity to advance the predictive understanding of watershed processes. So this special issue is I'm actually coordinating with Ron Popple and Ellen Wohl. So please consider this if you have anything exciting for this special issue. So without further ado, I want to talk about critical zone science. So in last couple of years, I have seen like NSF has been putting a lot of emphasis on CZ network, critical zone science, and also DOE has been doing a lot of that. And both the agencies actually use this term critical zone science. So when I was writing this paper here on the left-hand side, that we published a review paper in ERL. So in this review paper, we summarized almost like 500 papers, and that came out like from mostly from DOE funding from last 20, 30 years. So we have seen like how critical zone science evolved in last 3 decades. So it has started with microbiology, and then organomineral interactions, and slowly we moved in scale and complexity. And today, now we talk about ecohydrology and watershed science. So there are only subtle differences how NSF and DOE use these terms. And I came to know more about this when a reviewer actually asked a question, because we were using processes, they range from the bedrock to the canopy. So they said like, why don't you use like NSF type language that would be more general. So here we realize like although we do similar things, but some language is different. We use terrestrial environmental science, but both of these agencies emphasize on interdisciplinary research that integrates physical, biological, and social sciences. And also we talk about subsurface processes, biogeochemistry, and hydrology. So although I'm not going to talk about much on this paper, but I want to actually say, like, if you have time, go through this paper. So here, like in last 20, 30 years, what DOE has funded. So there are like 7 sites here, although this is like only a subset of critical zone science too. So there are 7 sites like Hanford, Riverton in Wyoming, Oak Ridge Reservation, and Savannah River site, East River, Rifle, Nevada. So all of these are our scientific focus area sites, and they focus on critical zone science. So I'm going to only talk about 2 sites here. Those are located in Colorado. So one is the Rifle site and one is the East River site. And these 2 sites, we have been working on this site for like very long time. Like Rifle, we have been working from late 90s. And East River, we started from 2015. And I think Praveen is very much aware of all this work because he was also on our scientific advisory board for our SFA. So he has visited East River site as well. So first I will talk about Rifle site, and that actually gives a rationale to move ahead with a scale adaptive approach. So Rifle floodplain, this is located in Colorado, and this was our genome to watershed scientific focus area site. And it was a uranium uranium remediation site. So here you can see that there are like naturally reduced sediments. Those are very fine and rich in carbon and sulfide. And then we have a lot of wells. I'll show where they are located. And we focused mostly on this TTO1, TTO2, and TTO3 transect. So in this site, like people have been working for like very long time, although most of the investigations were only limited to either 1D or 2D investigations. And here, Rifle site, so floodplain is always losing water. So Colorado is gaining a river. However, there are some instances when there is a hydraulic gradient reversal. So then here, Haruko, she with Flores, they actually did this naturally reduced sediment characterization. So we developed a full 3D model for this floodplain. And here I want to show a digital elevation model of this site. So you can see it's like relatively a flat site, like elevation difference is not like too much. And there are like several wells. So all these red dots, they are stilling wells. And we use them to constrain hydrologic boundaries of this site. And this green transect, and these are 3 wells. So TTO1, TTO2, and TTO3. These were like intensively sampled, and it was like very heavily instrumented site. And all these yellow dots, they are like wells where we collected biogeochemical samples occasionally. So this TT transect is also aligned with the flow direction. And as I said, like the Colorado is a gaining river. And note that this TTO3 is located within the naturally reduced zone. So here our overall goal was to evaluate how transient hydrological conditions control redox zonation and examine how hydrological flow paths, microbial activity, and sediment compositions influence biogeochemical processes. So we used pfloatran to develop a reactive transport model, and it's an open source, high resolution, 3-dimensional reactive flow and transport code. And Glenn Hammond is one of the primary developers of this model, and I think he graduated from UIUC. So you would probably know about this code. So here we used structured grid and Darcy-Richards equation, and then we used Van Newton parameters and Mollem formulation to relate capillarity and unsaturation. So here in this side, we represented two layers: fill, that was low permeability on the top, and alluvium, it was relatively a higher permeability region. And notice here we have these naturally reduced zones. So all the past site-specific studies actually focused on distribution of uranium and carbon hotspots. Not any of them like had like primary focus on nitrogen dynamics, and all the studies were either 1D and 2D. There is one study done by Steve Yabusaki from PNNL. So he developed a 3D model using ESTOMP, but he did not focus on unsaturated zone. But we did both the modeling studies like at the same time. So here we are using 3D model to understand nitrogen hotspots. So we also integrated a complex biogeochemical reaction network into pFlowTran. And here we had like heterotrophic and chemolithoautotrophic pathways. They were represented using Michaelis Menten kinetics. And then we had like several minerals. So all mineral reactions like precipitation, dissolution for like calcite, goethite, siderite, pyrite, and we represented organic matter as one single solid phase soil organic matter. So that drove this redox ladder. So we had aerobic respiration, denitrification, iron reduction, and sulfate reduction. And then chemolithoautotrophy led to sulfur and iron cycling. And this single solid, representation of organic matter that provided DOC to run this reaction, and we also had ammonium in the soil. So I talked about Colorado is a gaining river. So here I want to show like how it is actually when we look at the data. So in this figure, x-axis is time and y-axis is water level. So here you can see like this gray band. This is water level at boundary. So that arch-shaped boundary. So if we plot like all of those locations, so we get like this thick band. And then this river stage is like if you sample like each location along the floodplain. So that way you see like a river that fluctuates as this red band. So you can see like most of the times like so I would say just focus on these dotted green line and blue line. So dotted green line is water level at boundary, and dotted blue line is mean river stage. So you can see like a river is always like staying lower than the green line, except for these locations like mid-April to mid-June. So these are the times, and only like a few days, and it does not like stay that long. So when that happens, like hydraulic gradient reversal, so floodplain experiences change in redox conditions. So it stays anoxic, reduced for a very long time, but this gradient reversal actually changes that condition, and that leads to very interesting dynamics for nitrogen that we will see in next few slides. So to develop confidence in our modeling, we try to match water saturation and water levels. So here you can see, although we are not able to capture like everything, and it is because the kind of like instrumentation we had, so that also introduced some error. So, but it's still, this is like a fill layer and alluvial layer. That's why like we see like a change in gradient here. So here we were still able to capture this trend, and vertical stratigraphy is apparent here from the gradient change in the saturation profile because of fill and alluvial layers. So next year we try to compare like water levels from our model and observations. So these are like wells we did not use for constraining the boundary conditions. So here you can see like the model was able to capture hydrologic variability across the floodplain reasonably well. And one thing I want you to take a note, like all these wells, they had like very similar pattern. Like all the wells had like a rising and falling limbs, and this is like very much identical in all the wells, although water levels are a little bit different in some of these wells. from each other. So here we can also look at like hydrologic flow paths. So here you see like it was like gaining river. So most of the velocity vectors, they are like directed toward the river. But at 150 days, when this happens between like mid-April and June, when gradient reversal happens, so we see like velocity vectors change their direction, and it impacts the redox state in sediments. So we also looked at oxic conditions. So here on the left, we have modeling results. So here you see like before the flow reversal, it is mostly anoxic, but naturally reduced sediment that is like very, very anoxic. However, when flow reversal happens, so we are looking at like 19th July, it's around like 150 days, I think. So here you see like floodplain actually gets oxic, and naturally reduced sediment, it still stays like pretty much anoxic, but there is like some change in the contour of this naturally reduced zone here. And we compare this with observations on the right. So here you can see like observed DO concentration in groundwater suggests that river water infiltration results in oxic conditions for a very short time at TTO3. So then we looked at nitrate dynamics. So here x-axis is again time, and the y-axis is concentration of nitrate. And here we are looking at like wells in Wedo zone and groundwater. And this first column is near the hill slope, that is TTO1. And second is mid of the floodplain, that's TTO3. And TTO3 is near the river. And this is also located within the naturally reduced sediment. So here we see like TTO2 and TTO3, like they show a lot of variability. TTO3 here in Wedo zone. So these are like along the depth. So here when we simulated this floodplain, but we did not consider like rain. And especially we did not consider rain here because it's like very little amount of rain there and short episodic. So we see like we were able to capture only like average response. However, like when we included like rain, so we were able to capture a lot of these trends and nitrate values. And this nitrate comes from the nitrification from the soil. And this short and episodic rain event influence nitrate levels. And we also see like NRZ and non-NRZ like show distinct processes that control nitrogen biogeochemistry. So to confirm like if that nitrate is actually coming from soil, so we also looked at nitrate and chloride ratios in groundwater and Wedo zone. So here all red triangles, they are TTO1, and blue, actually red are in groundwater and blue are in red in Wedo zone and blue are in groundwater. And these are TTO1, TTO2, and TTO3. So you can see like in TTO2 and TTO3, like nitrate to chloride ratio is higher. That means like nitrate is coming from soil for those wells as shown by the model. Then we looked at how nitrate hotspots and hot moments actually migrate across the floodplain. So here I'm showing like 4 snapshots. So only like 150 days, this is actually the time when flow reversal happens. So from these snapshots, what we can see like hot and cold moments of nitrogen do not occur simultaneously. And when you looked at like flow hydrographs, you saw like it was like a similar pattern across all the wells. However, when we look at nitrogen hotspots, we see like there are like distinct patterns. So that means like only hydrology does not control nitrogen hotspots. It's actually biogeochemical heterogeneity that impacts them more. And spatial patterns of nitrogen species are predominantly driven by presence of these naturally reduced sediments, and to some extent by hydrologic flow paths. So once like we had this model developed and built enough confidence, and we were able to reproduce hydrology and nitrogen dynamics. So we also did like sensitivity analysis. So here on this view graph, you can see like top panel. So here we have like groundwater flow when it is typical scenario, like when the Colorado is a gaining river. And the bottom panel is when groundwater flow reverses. So this is like only mid-April to mid-June. And these columns, so here we considered all the reactions like heterotrophy and cumulative autotrophy. And on the second column here, here we considered only heterotrophs. So overall, like we performed like 24 simulations, and these are computationally very expensive. Like each simulation took like something like 250K CPU hours for 1 year of simulation. So this is like one of the reasons we need scale adaptive framework, because floodplain is like only like a small portion of a watershed. So if we really want to simulate like the entire watershed, it would be computationally very expensive. And here we can also see like these small scale processes like NRZs. They are so important because chemically reduced sediments of the rifle floodplain has 70% greater potential for nitrate removal than non-reduced zones. So when we want to model like a watershed, we have to incorporate impact of these small portions or regions like hollows or NRZs. So I will come back to that in my later slides as well. So I would like to summarize quickly like what we learned in the rifle site here. So we saw that nitrogen hotspots occur because of complex fluid mixing, localized to reduced zones and biogeochemical variability. And then chemically reduced sediments have more than 70% potential for nitrate removal than non-reduced zones. And then we also saw that 3D simulations were useful in capturing the significant spatial and temporal variability associated with nitrogen fluxes in the floodplain environment. So I would take a little break here and would like to ask you if you have any questions before I move to East River.
So Dipankar, what is the resolution at which you're doing the simulations? The spatial resolution?
Okay, this was like 6 feet. Sorry, I converted that in meter, but yeah, 6 feet spatial resolution. So that would be how much? Like something like 2-3 meters or 2 meters, right? Yeah, so it's like a few meters. And vertical was 0.25.
Okay. And this is the pfloat-run code that you're running.
Yes, yeah. So unless like we have more questions, feel free to ask. Other, you can stop anytime. I know Tarun told this is more like a informal discussion, so don't hesitate to stop me. So here now I will move to the East River site. So Ken famously said like life is too short to work on only a contaminated site. So here we are moving from a contaminated site in Rifle to pristine East River watershed. So here you can see in Colorado, this is Rifle site here, and this is East River. And all these green dots, by the way, they are DOE's like remedial sites where they conducted a lot of nuclear tests. So they are cleaning these sites. So these are actually described in that ERL paper if you are interested. So East River is a mountainous watershed similar to Rifle, but Rifle was only a floodplain. So here in East River, you can see like it's rolling to flat topography actually here in meander you can see. So here for this particular portion of my talk, I'm just going to focus on this small meander here. So this is an intra-meander region at the East River, and it has a lot of hybrid flow paths. So here you can see the scale, it's 50 meters. So from this upstream to downstream, I think it was like something like 60 meter or 62 meters. So it's not like a very large domain. However, we used like a very high resolution DEM data like 0.25, 0.5 meter. So even like we model like this small meander, that becomes computationally challenging. So here our overall goal was to quantify subsurface geochemical exports to the river system. So here, that's the DEM data I was talking about. So it's 0.25 meter resolution. And you can see like this high resolution DEM was able to mark all these small topographic features, high resolution features like here, gullies or like a small elevation. So this is the meander that we are modeling. And when we model this, like using 0.5 meter resolution using p-Floatran, so it becomes computationally challenging if we really want to do like a lot of simulations. And why I'm saying that, because for hydrology, it is okay. It's not that hard. It takes like, let's say for 1 year simulation, it may take like this meander with 0.5 meter resolution, like less than 1 hour, let's say. But when we include like all biogeochemical reactions and minerals and everything, and we have to spin this up at least for 40, 50 years. Although people say like, if you talk to Nick Switcher, he would say like, spin it up for more than 100 years. So when we spin that up, like with this kind of high resolution and all those mineral complexities, it becomes computationally very challenging. So here our scale adaptive approach was like we developed 2D transect model. So this is MCP transect. And other reason to use only this 2D transect, because we had wells where we collected all geochemical samples. So these are marked as like 1, 2, 3, 4, 5. So we call them MCP1 to MCP5. But there are other wells as well, like W1 to W9 or something. So these are stilling wells, and these are like wells actually baptized put there here. So one thing I also want to mention here, although I'm showing these are my papers, but a lot of people contributed to. So I want to acknowledge all my co-authors and other mentors who helped to guide through this work. So here, these are water level observations, and these are stilling wells. So Helen Melinda, she was a PhD, she was a master's student of Kamini Singha. She was working here, so she put those wells. And then we had these MCP wells. So we modeled similarly like the rifle site. We used p-Floatran, and I'm not showing all those details here, but we integrated a complex reaction network, same as like we did for rifle, although the reaction network was modified based on the local conditions here. So we also collected a lot of microbiology, biological samples, genomes, and based on that, we modified the reaction network. So here I'm showing, let me first clarify it a little bit. So here we used primarily this MCP transect for our main investigation. But at the same time, I also developed like these 1, 2, 3, 4, 5 other transects like equi-spaced. And then we lumped them, aggregated to capture the overall impact of my hydrology and biogeochemistry to get like what is happening in the meander. So we did not simulate like 3D meander, rather like we simulated like several 2D transect, and then we aggregated. So when we aggregated, so this is what we get. So here on this view graph, you see like DO, nitrate, iron, TIC, and pH. So here you see like DO and nitrate, they are like quickly removed as soon as like they enter the subsurface part, because it's carbon rich, and microbes like eat up like DO and nitrate quickly. However, iron shows like some interesting patterns. It shows a variability along and across this meander. And this is because like once like DO and nitrate is removed, then nitrate starts building up. And here there was a gully. I will show that in next slide. So because of that gully, like there was like some oxic recharge happened. That's why like it again became like iron was removed. And then you see like TIC, it actually increases along and across meander, because all the reactions, redox reactions, they are contributing to TIC. And pH has like pretty much like a homogeneous pattern. So here we see that intrameander hyperic zones display distinct anoxic and suboxic regions. And these suboxic regions are located along the sides of the meander band. So here, this is what I was showing, like gully was located here. So on this view graph, these are the locations on y-axis, MCP 1, 2, 3, 4, 5. And y-axis is like time. So you can see like different snapshots are like arranged in time in y-axis. So here you see like these are the times like when high river stage happens. So this is what like there is only like very small amount of time like when you see DO and nitrate inside the meander. Otherwise, most of the times like DO and nitrate are removed quickly. But iron shows very interesting pattern, and it is very much like how hydrology and biogeochemistry interplay. And this is like the gully here. So you see like oxic recharge happens, and then iron is again like removed from the system. And then TIC, it actually increases like as you move from MCP1 to MCP5. And pH is like does not show like a lot of variability here. So here gully is like, so you can look at like these small scale high resolution and topographic features. They have like a strong impact on redox conditions. So overall, this the transient hydrologic conditions impacted lateral redox zonation. Okay, so here then we also computed like net geochemical exports from a single meander using all these 2D transects. So we computed like how much is entering and how much is leaving. So this is like net export. So we see like TIC, DOC, iron and chloride. So here y-axis on the right, it is river stage that is shown in green. And these blue lines are redox species. So TIC. So here, what we see actually, meander acts as a sink for organic and inorganic carbon as well as iron during the extended base flow and high water conditions. And mechanisms are different, although it acts as a sink. The reason is like during high water conditions, river is not gaining water, so there is like less export. However, during extended base flow conditions, microbial activity is low. And also, I'm not showing that figure here, but it's in the paper. So when you look at the stratigraphy, so there is like a gravel layer on the top, and then there is like a clay layer, and then there is like a shale. So as soon as like water table falls and it is not in the gravel layer, so then flow becomes very low. So that's why like microbial activity, and maybe also because of temperature in winter. So that's where we do not see a lot of microbial activity. So in summary, like in East River, what we saw, intrameander hyperic zones display distinct anoxic and suboxic regions. And we also saw like permeability had like more impact than reaction pathways on biogeochemical zonation. Then we saw meander acts as a sink for organic and inorganic carbon as well as iron during the extended base flow and high water conditions. I will move to scale adaptive framework, but if you have any questions, please stop me. So based on these, yeah, please go ahead.
I have a question. I was interested for this model, could you do any kind of calibration, or did you basically have one set of parameters that you used and went with that because it was too expensive to calibrate the model?
Well, you can actually calibrate. There is no problem in calibrating. So I was using like 2D model, and partly that was the reason like we wanted to use 2D because we wanted to run several simulations. So when we were like developing these reaction networks, so we needed. So although even if you are using like 3D model, so what I do actually, I develop like also like analogous to 3D like 1D and 2D models, and use them for calibration, and then use those parameters in 3D model. So that's how you can do. But there is a postdoc working with me here, Lucian. So he has developed a particle swarm optimization method. Although he uses MATLAB, but that's also available in Python. So he has automated this calibration. If you are interested, we can talk about that after this talk.
Yeah, thank you.
Yeah, but it's like easily you can calibrate. 1D and 2D, I would say there is no problem. Okay, so based on like last two case studies, I would say, so what we learned, like we made like significant progress. We are able to capture high fidelity processes. Still, there are like several hurdles that we need to overcome to improve predictive capability at the watershed scale. And that is like, one is like processes. So here processes, they range from genome to watershed scale, and they are all linked and non-linearly coupled. And most importantly, local fluxes of carbon and nitrogen influence downstream water quality. So even when we do like watershed scale simulations, we have to respect local fluxes, and we have to represent processes accordingly. And second is like multi-scale, multi-physics watershed capabilities are needed. So a lot of times, like when we do like watershed scale modeling, so we refine like close to river, and then we coarsen. So we need to have like proper model structure that can represent the system reasonably well. And it's not like very well understood. Can we use like effective parameters or different parameters? So that's like one of the challenges. And finally, computational inhibitions, like scaling process-based simulations are computationally expensive. So even if we have like supercomputers available, it's not tractable. So to address these problems, our goal was to develop a scale adaptive framework that combines process fidelity with computational tractability. So here we wanted to compute geochemical exports and river water quality as the function of characteristic watershed features like topography, wetness index, meanders, sinuosity, and amplitudes. So here we developed a scale adaptive framework that's actually a cascade of process integration from meanders to, let's say, floodplain that includes the 10 meanders, then sub-watershed and watershed. So here, this is the same site like I showed in my previous case study. So East River watershed. And then we divided this watershed in like different segments. So here it's the lower East River watershed, and then here the floodplain that has like only 10 meanders. So here you can see this is the region where this 10 meander domain is located. And then we use like 2 meanders to develop scaling relationships. So first we developed a p-flow model for these 10 meanders. And this is still, I'm still working on this paper. So this has not been published yet, but I'm hoping to probably submit this paper sometimes in next few months. So we again use p-flow and developed this model using structured grid and Darcy-Richards equation. So here we imposed like a boundary condition on these river like Dirichlet boundary conditions, that like fixed head boundary condition that changes in time, and hydrostatic boundary conditions on these sites. And we had all those wells I showed in a couple of slides ago. So we used those wells to constrain these boundaries. And then we also had like heterogeneity. So like floodplain had like different stratigraphy and different parameters that we took from other people like Bhavna and Lucian. They developed floodplain model, hill slope models. So very detailed hill slope models they developed, and Lucian published one paper in GCA. So if you are interested, you can look at like, he did actually deep time weathering. So you can look at like how detailed modeling in 1G he did. So I took like borrowed parameters from Lucian and Bhavna. And for floodplain, I took most of the parameters actually from my previous paper that I showed before. And then we developed also like some intermediate products like sequential imputation method to fill gaps and constrain these boundaries. So the challenge was here, like p-flow is like only subsurface code. It does not solve surface flow. And there are other codes like HCS Amanzi, they can actually do surface and subsurface flow, but they are not very good with biogeochemistry. First, they use like a peak Flowtron or Crunch with alchemia interface. So that becomes like computationally more challenging to do like this kind of detailed biogeochemistry I was interested in. And second, they do not do biogeochemistry in the surface flow. So because of those two reasons, I actually developed a river Flowtron module and integrated this fully dynamic 1D shallow water equation in P Flowtron. And it's a mass balance equation. It takes into account momentum balance, local acceleration, hydrostatic forces, gravitational forces, and roughness forces. Actually, so Ilhan, he was a postdoc here at Berkeley. So he was working with me on this. So most of the integration Ilhan did. And now he is faculty in Germany. So this river Flowtron module, what it does, like if you look at this, like this is a river cell, for example. So it takes in flow, it solves flow, and then we solve like biogeochemistry for each river cell, like as you go along this flow. And then we sequentially couple this and solve biogeochemistry, and then take into account like what goes away from one particular cell and what comes into, and then we solve chemistry and flow both for this river cell. So that's how we can actually account for bidirectional exchange of geochemical species and quantify changes in river water quality. So I'm going to only focus on 2D, two meanders. So 10 meander work is still going on, but we used this river Flowtron module and simulated this to these two meanders. We took a subset from those 10 meander models. So here we can see that East River is a very highly dynamic system, and you see like flow velocities, they change, and this river Flowtron model was actually benchmarked using some analytical methods. I don't remember the name, but there was like one paper that actually developed like some analytical methods for those surface flow equations. And so here iron, you can see like it's like again like very it shows a lot of variability, and nitrogen also shows a lot of interesting patterns. You can see these footprints of meanders, and then with high water and low water conditions, nitrogen and iron, they have like very different signature here in the floodplain. So we also looked at like hotspot and hot moments of nitrogen in this river when we simulated using river Flowtron and P Flowtron models. So here we see that these are the locations where we always get like irrespective of low water or high water conditions, we always get like hotspots of nitrogen. And they are produced due to river stage, bathymetry, and meander geometry. Let me show like what I mean here. So if you look at like these two, I'm talking about these two meanders here. So when we integrated this river Flowtron model into P Flowtron, so this was one advantage like using the structured grid. So here we can have like these multi-directional exchange. So here, like some cells, they had like only north-south exchange, and some cells, they had like only east-west exchange. But some cells, they had like both east-west and north-south. And this was based on like bathymetry and river stage. So here we see that cells, those had like east-west and north-south exchanges. They are more active biogeochemically, and hotspots of nitrogen are formed mostly mostly in those locations. So then our next goal was to develop like a scaling relationship. So we took this two meander model, and then we looked at like amplitudes of meanders. So like looking at their sinuosity. So the goal was like if we can actually relate with amplitudes and sinuosity, so we might be able to predict these hydrobiogeochemical exchanges across the river based on sinuosity and amplitudes. So we simulated like 4 different scenarios. So we had like amplitude 1, 2, 3, and 4. So this is like the highest amplitude, and this is lowest. And here you can see this is highest amplitude, and this is the smallest one. And I'm going to show like some simulations based on these scenarios. So when we looked at like denitrification potential from these 4 scenarios, so amplitude 4, and this is the smallest. So we see like with larger amplitude, you have like more denitrification potential. And these colors, they show like rising limb and this reddish is recession limb, and green is base flow. So you see like recession limb and base flow, they are like have like some interesting relationship based on amplitudes. However, amplitude has like a very significant impact. And we tried to explain this, like why this is happening. So we looked at like all those reversals. These are like reversals. I think they were like 1 to 72. And this is the flow direction is like from top to bottom. And the x-axis is time here. So we looked at like reverse stage in all these cells. And we see like there is like some pattern. They actually control like number of neighboring subsurface cells. They are submerged. So it's actually an interplay of like how many cells are submerged, and like what kind of exchange, northwest or east-west, or like it's both synchronous exchange. So they all actually play together and control denitrification potential of the floodplain here in the East River. So this is my last slide. I would like to thank you and leave with these conclusions, and take any questions if you have.
Thank you, Dr. Dwivedi, for a very interesting talk. I was hooked from the beginning to the end. The floor is open for questions.
Well, I don't have a question, but I have a conversation with my colleagues, Neil and Andy. I mean, this seems very apt for the kind of work we are doing with the Allerton area. Is this an opportunity here to work together to do some simulations of the floodplain?
Can you hear me okay?
Yeah.
Yeah, so yeah, you took the words out of my mouth, Praveen. So because you know, Dipankar, you're getting to the things I've been thinking about, but not knowing how to quantify things. So we're looking at both dissolved species and particulate species and how they behave on the floodplain. And the thing that I've been trying to get my head around is the relative importance, or how overbank flow has an impact on like particulate delivery and chemistry versus the groundwater flow that's coming in and basically flooding a floodplain from the bottom up almost. And it strikes me you're getting dangerously close to be able to address those things and put it in a model and test some ideas. But anyway, let you comment on that. See what have you thought about that?
Yes, so that's actually a very interesting comment. I mean, we do not see like that kind of flooding, or maybe we see, but it gets actually frozen. So yeah, so we can use actually seepage phase boundary condition and simulate that kind of behavior.
So we have, thanks to Molly Kane, who's a PhD student from Indiana. I guess, I don't know if she's still a PhD student, or has she, I guess she's defending soon, who has a large number of wells out on our floodplain, and I think has done a nice job of mapping over time the flooding sequences and where the water flows, which strikes me would be hugely valuable for this type of model to initialize it.
Totally, I would agree, and it would be interesting actually for different hydroclimatic region to do similar modeling.
Yeah, and she'll be giving a talk to us on the 14th, actually, of her research. She's going to be graduating, or at least attending her graduation in May. So,
I
but
have
we're
a, oh,
but,
sorry, go ahead.
but we're interested in like knowing also how the meander channels interact with the groundwater and the surface water, because those seem to be active more of the time than the floodplain, most of the floodplain. So how those interconnect with both the surface water and groundwater would be of interest in a three-dimensional kind of situation.
Right. So yeah, so I haven't actually shown, like we also developed like a module like particle tracking. So that actually can visualize, I mean, it's like nothing like very fancy that way, but it can actually visualize like how one particle enters like from upstream side and goes like how many times in the subsurface and then resurfaces the stream water. So that also actually impacts like how much biogeochemical activity or hypericity exchange that happens in the system. So I totally agree. Like that's very interesting thing we can look at.
Dipankar, when your group, I mean, the bigger group that's been studying these DOE sites,
Mhm.
you know, you've specifically have focused on dissolved species for obvious reasons, but what's been done with looking at the solid phase matrix, geochemistry, and where I'm going with this is that we want to look at the weathering of the so-called soil or the sediment in the floodplain. And thus far, when we look at a lot of parameters, this is something Allison's been doing. She's having a hard time seeing vertical changes. And I'm wondering if that's a problem with our perspective, in that with so much lateral action going on, that we're maybe using the wrong axis to look for weathering. And I'm just wondering what your experience has been on this.
Yes. So here I shared like one paper on weathering that came out in GCA. So Lucian, he is a postdoc with me. So we collect like a lot of sediment samples, and I'm not like an experimentalist. Sometimes I go to field for learning that, but I'm not primarily a lab person. But we collect a lot of sediment samples and do solid phase analysis. So here, Patricia Fox and some of those guys, Wenming Dong, they do. So we characterized all like different minerals, and they do XRD and some other fancy things. So that tells us like what kind of solid phase is in there and how to represent. We talk to them and discuss like how to represent them in our models. So yeah, I mean, but it's important. I totally agree.
And then yesterday I was reading an article about, I guess it was about coal mine waste, but there's this perception that the Midwest is getting more rainfall or more precipitation, so that means that the groundwater level is going up. That's at least what they were concluding, but because there's more rain, but that may be not the case, because there's maybe heavier precipitation events which may not infiltrate the subsurface. So be interesting to validate that assumption that more rainfall equals higher groundwater levels.
There's a question in the chat from Lisa Webb. So she asks, for the Rifle River site, you showed nitrate concentration increases during oxic events. Can you comment on nitrate removal? Can you comment on how nitrate removal was quantified in the model during reduced conditions, and how does a zero nitrate concentration inform the rate of removal?
Okay, so we were looking at like this 2D transect, And so we computed like how much was actually entering the river. So looking at like that interface, so that's where like we computed like nitrate removal. And zero nitrate removal, I don't know, like because we were looking at like the annual time scale, so it's like not instantaneously. So I don't know like what zero nitrate removal would mean, but I don't know if that answers your question. So we looked at like in 1 year, like how much nitrate actually entered the river. So that was like because it's a mostly a gaining river, and only like few days it's a receiving river. So everything was like going from floodplain to the river. That was considered probably removable.
Thank you. Do we have any more questions? All right. Let's thank Dr. Dipankar Dwivedi once again for giving us the time to give us the talk, and thank you everybody else for coming and attending the talk. So have a good rest of your day.
Thank
Thanks,
you.
Dipankar.
Thank you, everyone. And please feel free to reach out to me if there is any interest in collaboration or anything. I mean, I will be happy to work with you, and I can also spend time in teaching free-flow trend modeling if some new students actually want to try.
Thank you.
Thank you.
Thank you for the talk.